Hey guys, we're going to go over really quickly the process of protein synthesis. I want you just to have an overview of the process. You've been practicing the last couple of days on transcription separately from translation, but this overall process is really kind of the big deal in biology. It's like everything works because of this process, okay? This idea of protein synthesis. Making proteins, right? Everything runs on proteins. All of the enzymes in our bodies are proteins, and we know that we couldn't do those chemical reactions without them. Everything is made up of proteins. Proteins also dictate the phenotypes that you see: hair color, face color, your intelligence level, your abilities, any diseases, anything like that is all dictated by the proteins that your body makes, and that is all coded by DNA. Okay, so the first part of this process starts in the nucleus. So this black line here is showing you this side is the nucleus, that side is out in the cytoplasm. First you start out with DNA in your nucleus. RNA polymerase is going to bind to your DNA strand, open it up, and start coding a segment of mRNA. Now, why does this happen? Why doesn't the ribosomes just use regular old DNA? Well, the problem is you want to keep your DNA safe. You don't want to get it out of the nucleus. It's safe and sound in the nucleus, so you don't want to let it out into the cytoplasm to get damaged. So basically, your DNA is like your original blueprints, right? Your nucleus is your file cabinet. This is your original blueprints. But when you have an electrician or a plumber come, you don't want to give them your original blueprints. You're going to make a copy of your blueprints, send that out with the electrician to go put in some electricity, okay? And so that's kind of what's happening. This mRNA is going to be a copy of your blueprints. So once your DNA strand opens, RNA polymerase is going to read the DNA strand in the 3' to 5' direction. It's reading it in that direction, but it's building an mRNA strand in 5' to 3' direction. Okay, I'm going to make another video to make this idea a little bit clearer, because it looks like some people were having some confusion. So I'll explain why this is important, why things get built 5' to 3'. But it's important to know the difference between these two. These are anti-parallel, and the RNA polymerase is building it in the 5' to 3' direction, okay? Now, this part doesn't make a whole lot of sense. The part that is not being used by the RNA is called the coding strand, or the sense strand. The part that is being used by the mRNA is called the anti-coding strand, or the antisense, okay? It seems like it would be the other way around, but basically what's happening is that this mRNA that's being built matches this one. So that's why it's the coding strand, because this matches what is being built over here, right? Because this is going to be the opposite of this. So if we're building one that's the opposite of this, it's going to match this. Does that make sense? So you got to kind of wrap your head around that one. This is what we would call pre-mRNA, because it's not mature yet. There's some things that have to happen to it in order for it to leave the nucleus. So we're going to put a GTP cap here with some guanine. That's going to kind of just protect it and stabilize it. Both the cap and the poly A tail are going to stabilize it and allow it to stay together as it leaves the cytoplasm, or leaves the nucleus. There's also things called introns and exons, and introns are like the junk protein. Okay, we're going to call these junk. The junk code, and we're going to get rid of those. It's a lot easier to read when you get rid of the gobbledygook, and sometimes there is. So this is also when this happens. So before it leaves the nucleus, the introns are excised out. The exons are the ones that remain. And then once this is all modified, it is now called mature mRNA, and it can leave the nucleus. So then, this is the second part. This is translation. And this happens at the ribosome. So you have your mRNA strand leave the nucleus, you have a ribosome bind, you have a large subunit and a small subunit bind around the mRNA. And what happens is there's usually some code on the mRNA strand that is a initiation code, and it's a code that says start coding here. And so you're gonna have a tRNA. So these ones are tRNA. And tRNAs carry with them amino acids, and it's going to match. So here's the codon on an mRNA strand. This is what we call an anticodon. It's basically the one that matches it. So a codon is a series of 3 nucleotides. Okay. So every 3 is a codon, and it's a code, it reads it. So if this is UC, because remember it's RNA, so it has a U instead of T. So U binds with A. We remember A and T bind together. Well, if we're talking about RNA, we're talking about A also binding to U, and C still binds with G, that rule still applies. Okay, so these are our base pairing rules. So U is going to bind with A, U binds with A, C binds with G. So when this matches this code, it will bind, and they'll both kind of like sit into the ribosome, and then the ribosome moves, okay? Once a tRNA binds, the ribosome moves down this way, moves down this direction, and then it goes to the next codon, and then a new tRNA combines, and then it goes to the next codon, and then a new tRNA combines, right? And there's things up here that once the new tRNA sits here, it binds, excuse me, it binds the incoming amino acid with the growing polypeptide chain, okay? So this is the protein that's being built, and we're calling it a polypeptide chain. Now, obviously, proteins get a lot of their function by their shape, their tertiary structure. And so after they're released from the ribosome, then they're gonna fold up and form the shapes that they form, okay? So right now we're just seeing the chain of amino acids, and it'll keep going. This process is called elongation because it's where the polypeptide chain is getting longer and longer. And then we have termination. Once it gets to a code in the mRNA that is a stop codon, it'll tell the ribosome to stop coding. It'll signal a release factor to come and bind, and that release factor will allow the polypeptide chain to cleave off and go about its way. It's gonna go be processed and fold up in its orientation and do what it needs to do, and then the ribosome's subunits will leave, okay? So that's the whole process from transcription, where we're taking DNA, making mRNA, modifying mRNA, finding a ribosome, allowing tRNAs to bind and bringing with them amino acids. That continues until we have our whole chain of polypeptides, and until finally it releases, okay? And that's basically it in a nutshell. I know there's a lot more steps, a lot of steps, but if you can kind of look at it in a more broader sense, hopefully that helps you out a little bit. Okay, guys, take care.